Renata asked me to give an update on the phase 2 of the National Honeybee Survey. So I'm just going to mention a little bit what the difference between the last survey and the new survey that we are proposing. So we have been presenting to all the provinces the National Honeybee Survey that we run from 2014 to 2017. So the first phase of the survey was to determine the incidence and distributions of pests, pathogens, and parasites that affect honeybees in Canada. Also to test for the presence and absence of exotic threats. So it was funded by Growing Forward 2. The Alberta beekeepers were leading that project, so there was a cash contribution from Alberta beekeepers, Manitoba beekeepers, and CropLife. But we sampled from a lot of the provinces, but was led by Alberta beekeepers. So the target was 0.5% of the hives in Canada, and the distribution of the number of samples per province was according to the density of hives. So a lot of the samples, most of the samples were sampled in Alberta because most of the hives are in Alberta. We had a few inspections, so if you guys remember when we presented, so we have technicians that go to the beekeepers, to the operations. They will inspect the hives. So we target yard samples, but we inspect at least 10 hives in each yard. Sometimes we need to inspect more because we only collect samples from queen right hives. So some of the yards, if the hive that we're inspecting is not queen right, we need to open another hive and collect samples. And we test for the most common pathogens and parasites that affect honeybees. So Nosema, Varroa, Trichomoniasis, American foulbrood, European foulbrood, 8 viruses. We also, in the last year, we test for neonics in bee bread and exotic threats. That was threats that are not in Canada or expected to not be in Canada. So Laelaps mites, Apis cerana, Africanized honeybees, and that kind of stuff. So we test for that. So if you guys remember, so I'm not going to go over the whole presentation again, because every year we gave an update for those 4 years of survey. So we sampled 944 samples, yard samples, during that period. And if you see the map there is a number there in each province. Those are the number of yard samples that we collect. So in Alberta was 517 yards that we collected. So in each yard, again, was 10 hives inspected. So we inspected around 9,500 hives during that period. And was, as you can see, so Canada is a very large country and it's very challenging in terms of logistics to coordinate with all the technicians, the sampling. We had a lot of help from tech transfer teams and some of the provinces. So BC, for example, a lot of the samples were taken by inspectors, so that was good because they are very experienced in that. So the province gave that in-kind donation to the project. The Maritimes tech transfer team there also helped collecting samples. And in other provinces, we had to hire technicians to collect for us. Alberta government, so Shelley Hoover and Linné also helped quite a bit in collecting samples for us in the south there. But as you can see, it's a large country, there's a lot of challenge. And as you may notice from previous presentations too, so we didn't have any samples from Saskatchewan. So one of the problems of the project was not all the provinces were on board of the national survey. So we had a few challenges with that. With the new survey, so we are applying, to a second phase of the survey. So some of those challenges too, to get all the provinces on board, because some provinces, they agree in some of the tests, the analysis to be done, and some they want something else. So it's difficult to have everybody agreeing what we need. We have been working with Rod Scarlett from Canadian Honey Council. So we're going to apply to the Canadian Agricultural Partnership. So there's no Growing Forward 2 this time. So we're applying through the stream of agri-assurance programs. So it's the stream that have for the National Industry Associations. So that's why we are working with Canadian Honey Council. And Rod had a lot of work with all the associations in different provinces. And we got an agreement from the provinces. And also discussing about the cash contribution, because this stream, 75% of the money comes from this program, from this federal program, but 25% of the cost of the project need to be cash contribution. and also some in-kind. So we have been working with the provinces to see the amount of money that they will contribute, as well as how much in-kind. So a lot of provinces that have the tech transfer teams, they will be helping us, so as an in-kind contribution that has also a cash value because it's their time to collect samples. And as part here, just highlight there, so as covered by this, this program, they do have surveillance for animal and plant health. So that's why we decided to pursue this program. So the national survey, so the phase 2, the target is to have 350 samples national, so from all the provinces collecting samples, again, proportional to the number of hives. And at the end will be around the 0.5% of the hives in each province. But here we did some epidemiological study, and we used the provincial epidemiologist to help us with the survey. So we are using Varroa as the indicator. So based on the data that we got from the survey, that we found that around 70% of the hives in Canada were infected with Varroa. So we used that in the calculations there in terms of epidemiology. But again, it was very similar to the 0.5% of the hives that we need to collect. So what is new? A lot of provinces, they say, okay, last survey we sampled in the summer, so it was July and August. And there was a lot of provinces that they would like to see some data in other time sampling. So we decided to add a temporal study, so collecting samples in other times. So it's going to be in the fall, after winter, and then early summer, and then we have the late summer samples. So we are including more samples, but it's going to be a subset of the yards or the beekeepers that we are working. Again, so it's going to be voluntary participation. In Alberta, we had our target of samples. We had to say no to some beekeepers, especially in the Peace. We had a lot of beekeepers that want to participate, but we couldn't take them all in the survey. So again, with the help of the epidemiologists and the province, Dr. Dolores Peters there, we select a system, so it's going to be a random selection of the beekeepers in the different regions, distribution of the regions there, as well as beekeepers have different yards, so we're going to have randomly select as well the yards that we're going to collect, and again, have another plan for the yard, which hives in that yard we're going to select. So it's going to be based on epidemiological methodology to do that survey. Due to the importance and the risk of the exotic threats, we are still going to keep at least for 1 year on the project to test for Tropilelaps, Africanized honeybees. Those are very important to take in consideration because of the risk in terms of trades and other risks that we have. So at least in 1 year there, we will sample. If you guys that participate in the survey know, Tropilelaps is a lot of work to test for that. So we have that knock test that we based on the methodology developed by USDA there. So it's very labor intensive to collect for Tropilelaps. So we need to knock that frame, collect the debris, and then in the lab we need to screen that debris under the microscope there and look for Tropilelaps. So it's very labor intensive, but I think because of the risk that those threats have, if we need to deal with that, we will include them there. Excuse me. We also left for outbreaks and emergent disease. So if in the budget there, if there is a new disease that affects honeybees in Canada, we have on the budget there to deal with that. So we know there is emergent pathogens. For example, Critidia mellifica and Lotmeria passin. There are trypanosoma that before we were not talking about them, so we know they are here and what they are causing. So there is a few research ongoing there, and Courtney, it's working her PhD and using Lotmeria as well. So we want to include those new pathogens as well. And of course, the common pathogens, so Nosema, Varroa, and American foulbrood are very important. We also decided to expand the chemical residues. So last time we only sampled and analyzed the samples, the bee bread samples for neonics. So this time we would like to check for other other agricultural pesticides, but as well as miticides, antibiotic residues, and fungicides. So we want to rotate, but check all the samples for that because I think it's very valuable. So with that, so we think that this information that is generated by the National Survey, it's going to build a robust database. We have all the samples that we collect. We do have a large biolibrary in our lab there. So we do have bees that we sample across Canada, and they are still frozen in the -80 there from all those samples, because sometimes we never know what we're going to need those samples for, if another lab will request samples or pathogens. So we do have a collection of all the pathogens that we analyze. For example, American fall brood, all the American Bacillus larvae strains that we were able to isolate in the lab, we do have in glycerol stock, so we do have a huge library of those pathogens as well. It can be used for research for other purposes as well. So it's very important, and it's similar what other countries have been doing. So in Europe and US has so many years of the survey there. So we, the last survey, the last 4 years were the first time that we had that standardized survey done. The same methodology used in all the provinces. We know that different provinces, they do have some surveillance and they have been ongoing, but it's not in the same standardized system using the methodology. Also to use the data, the results as a reference point for emerging disease, as well as the pathogen pattern that we have. So I think with that information that is very valuable to use, and bee management, so we're working with province and policy making and decisions, and to know, so we have been talking here about knowing your enemies, so know what pathogens, what the distribution, what pattern we have the pathogens in Canada is very valuable. And for the beekeepers to apply, so it is a 75-25%. I think it's a very good investment if you think in that way from the money from the Beekeeper Association to put towards this project. So some of the results, so I'm just going to mention here, I have a few slides that we have presented before just to show a little bit the value of the information that we have from the survey. So we talk about Nosema infections, so not just here, we don't have a graph showing the spore levels, but we did check the spore levels for the samples in all the years there, and we report to the province. So the data is based on each province and the geographical regions in each province. So for example, here to know the composition, what species of Nosema we're dealing. So throughout the years, we analyzed not just the spore level, but the species. So we have been talking about, nowadays it's Nosema cerani. We know there's some research and some testing done in different provinces, but to have that empirical evidence that the main Nosema species that we have been dealing is Nosema cerani. So with this data we do have here, so that yellow part of the graph there, it is the percentage of the samples where we detect only Nosema cerani. So we know that Nosema cerani is the most prevalent species of Nosema in the samples, and you see the orange reddish part of the graph there is Nosema apes only infection. So we know it's very rare. So beekeepers that do have their capabilities of checking for Nosema spores in their operations there, we know that most of the Nosema they are dealing is Nosema cerani, and we know there is other research and trying to identify the differences between Nosema apes and Nosema cerani infection. We know that doesn't cause that dysentery that Nosema apes cause, so there is some biological differences there. So knowing the enemies that people have been talking about, so Nosema is mainly Nosema cerani species. Varroa, as we had a lot of talks about Varroa yesterday. So knowing the Varroa levels here, so we do have throughout the years the percentage of samples that we analyzed we found Varroa. So in 2016, we know almost 85% of the samples, the yards, there was Varroa in those samples. And those samples that we collected, graph that I have here, to emphasize again, those samples were taken in July and August. So you see that in 2016 there was almost more than 2% infection, so more than 2 mites per per 100 bees, and that's in the summer. So we can imagine as the bee population goes down in the fall, the mite population goes up. So that's an indication that we will have a big problem in the fall with Varroa, because it's going to be higher than. So, and as was mentioned yesterday, to know before it's too late in the fall, to know that the Varroa infections may cause problems. Even when we are able to control and kill, they cause problems in the hive there. We know that the viruses that they carry and transmit will be in the system. So even if you remove or control the Varroa there is some impact in the colony in that way, because the viruses are in the system there. So that data, so we do have for every single province. Another data that I think is very valuable is American foul brood spores. So we did check all the samples. So not just the inspections and seeing if we see symptoms in the hive, but we collect swabs if we see to confirm the infection, but also we detect spores, American foul brood spores in adult bees. And detecting the spores and growing the Penibacillus larvae, we could do antibiotic resistance. And that, I think, is a very valuable information there, especially with the federal plan to control antimicrobial and antibiotics and the antimicrobial resistance. So we know that most of the oxytet resistance case of Penibacillus larvae are concentrated in Alberta. There was 1 year that we found one case in Manitoba, but we know what we're dealing here. There's a lot of cases in Alberta that are Penibacillus larvae, so American foul brood resistant to oxytet. And that kind of information helps us how to manage and what kind of decisions we're going to make, and to adapt to the new federal plan as well. What was very valuable, so we used here the methodology and the thresholds that were used in research. So based on Dr. Steve Purnell's work and others there in Beaver Lodge, were with a sample of adult bees. So if you go to the hive, collect a sample of adult bees, we can analyze that sample for American foul brood spores. And based on the number of spores that we can grow in those Petri dishes, we can give a nominal risk of that hive or that sample developed active infection of American foul brood. So if you have a few, so we use between up to 99 CFUs, so spores there per bee. That was a possible risk, so probably not going to see an active infection. But if you see more than that, between 100 and more than 1,000, some of the cases that we found, definitely you'll find an active infection. So those are information that you can use to manage. So you don't need to even open the hives and inspect every single frame and look for the diseases, for the symptoms of the disease, but you can collect a sample of the adult bees and check for that. What is the risk of you developing that? So it's a way to monitor your hives and know the levels of spores there, especially if you're using an antibiotic in the past, where a lot of beekeepers were using prophylactically. We may not see the symptoms, because antibiotic will control the bacteria there, but it will not kill the spores. So the spores will be around, especially in honey, that you may have a reservoir of those spores, because antibiotic doesn't destroy the spores. So you may have that reservoir there. So that kind of information is very valuable, not just for the health of our domestic stock. And I don't know if there's any beekeeper here exporting honey to China. So the Chinese request that the honey exported to China to be American foul brood spores free. So we have been working with some provinces to test the honey from those beekeepers. So the province will do a field inspection, a visual inspection. We also receive samples of the honey. We test and see if we can detect. And then with that information, if it's free of American foul brood spores, the provinces that we have been working, especially Saskatchewan and Quebec, they will issue a sanitary report. So that will be a report used for the export permit. So that kind of information, I think it's very valuable as well. Viruses, so we talk a lot about viruses, the relationship with Varroa. So with the survey there, we found that the most common viruses, the most prevalent viruses in our hives are black queen, deformed wing, and sac brood. We didn't test for deformed wing B in this survey, but we know it's very common as well. So the VDV, or deformed wing B virus. Acute bee paralysis virus, was the less prevalent. We found very rarely in the hives, and it makes sense, it's acute. So the bees that we're collecting are live bees, they're inside the hive. So we didn't collect the dead bees in the entrance or bees that died. So was not the best sample to detect acute because it kills very rapidly the bees. Another virus that we tested in one of the years was Lake cyanide virus. So it's a new complex of virus. So we test from strains 1 to 4. And it was very common as well. We detect in every single province. We don't know exactly what it caused and the symptoms there, but we know that it's another virus that affects honeybees. Other information that we gather, so here's one example. We checked the viral infection in migratory and stationary beekeepers. So we did that also. We found some data there, some difference in the Varroa levels and the Dosima levels. But here is one example for viruses. So we know there was differences between the migratory and the stationary operations for Israeli, Kashmir, and Lake Sinai. So that information we also got with the survey. So when we asked beekeepers about those colonies, if they were stationary or migratory, and then we were able to compare. Exotic threats, so we analyzed for Tropile labs, Apis cerana, and slow bee paralysis. So those are threats that we don't expect to find in Canada. So it's kind of disappointing sometimes to run those tests because you know it's going to have a lot of zeros there and negatives. For those that work in the lab, you know that when you don't see a band or a peak, or it's kind of. But it's very important. So why we are testing this? We think they are not here because we need the empirical evidence. So yeah, they are not here, but are we looking for those? Did we have a system? Do we have a methodology? Did we survey? Until the survey, we didn't have that empirical evidence. And that it can be used to support the Canada sanitary status for trade of bees. So we can prove that we have run the survey and we do not have those threats in Canada. Africanized honeybees. So we did test the bees in 2016 and 2017 for using two different methodologies. So we used the mitochondrial DNA. So we analyzed the samples in 2016 and 17. Around 8% of the samples we tested, we were able to detect some ancestry from Africanized mitochondria there. And that's a test that is recognized by CFIA for the trade. But we know there is some problems with the methodology. So we tested and we asked different labs as well. So the lab in Guelph to test as well for us to confirm. So it was confirmed that was Africanized mitochondrial DNA ancestry there. So we know there's problems first because it only checked the maternal content of the genetic content from the queen. So it doesn't take into consideration the drones. We know that there is that problem, and we know there is some level of ancestry, Africanized ancestry in the mitochondrial DNA there from the evolution side there. So we know that there is some. So we found 8% of the samples. But when we run a different methodology that has been studied by Emerald Zayed in New York University, so he is developing the markers for Africanized honeybees there. So we test our samples and all the samples, they range between 0.6 and 16% using the whole genome of the bees there that were Africanized. So that's below the threshold that he has been established. So to be considered Africanized honeybees, you need to have 25% or more of your genome to be Africanized there, related to the Africanized honeybees. So we know that the samples, even though we detect some mitochondrial DNA ancestry related to Africanized honeybees, using the genome, so that's including maternal and paternal, we did not detect. So with this survey, it's good that we can develop new techniques and use new techniques that are improved to test the Africanized content in the bees there, the genetics. So we check also for chemical residues. So in this case, and we use 1 year bee bread samples from the hives there to test for neonics. So here we have a graph, and it cut a little bit on the bottom there, my sentence, but we detect, and they were very, the levels that we found in the bees, they were below what is associated with sublethal effects of neonics. But what is important here, as that pie chart shows there, is that more than 50% of the bee bread samples that we collected, we were able to detect 2 or more neonics there. So when we talk about the synergy, so this is just about neonics, so how many other compounds? So we know that in the past we have speakers talking about the chemical residues in the beehives there. So just in terms of neonics, we know that we found 3 or more neonics in the bee bread residues. And that's why we want to expand our analysis to other chemicals as well, to know, even though they were very little levels there below what we consider the threshold, what is normally associated with sublethal effect. So the level was very low, but we know we find a lot of those residues. And when you think about the synergy of neonics with miticides, with antibiotics, with fungicides, so we know that the level of the detection there, it might be amplified when you have the synergy between a lot of those compounds. So that could have an effect on the health of the bees. So those are some of the graphs and some of the data that we collect in the 4 years of survey. We have been working with the program there. I know they have a few questions. The officer from the program, and they call the honeybee industry the honey-making industry. So you know that they are targeting a product, honey, and that they want to see the program or the money used for honey. But we know, you cannot take the honey and separate the health of the bees. So you need healthy bees to produce honey. So we are just changing a little bit, adapting our proposal there to use some of the words and telling the story that they want to hear as well, because they are focused on honey. As you see, we are not testing only honey, so we are not collecting honey samples. That is complicated there. There's no honey in some of the sampling times. You only have like brewed honey samples. So we are working with the funder there to make sure that they understand that the honeybee industry is more than honey, that the health of the bees, that some beekeepers their main focus are not produce honey, they're pollination. So the honeybee health is more than honey, that the pollination service and the value added to other crops because of the honeybee pollination there, it's very valuable. So just to make them understand that a little bit. So we have been working with Rod back and forth with that. So we think that the continual effort of this project will benefit the industry, not just the beekeepers that are making honey, the honey making industry. Also, we are making sure that the funders understand that this will affect Canada, the community in Canada in a multitude of levels. So it's not just the health of the bees, but it affects the consumers. So the honey that we consume, and the suppliers and the trade, so the honey that we export. So giving that value and confidence, and for the consumers, knowing that the type of product that we produce and that we consume internally and we export. And just to give a segue to what Renata is going to talk about as well. So with this data here, we think we can work closely with the provinces, so the provincial apiculturists and the tech transfer teams in each province. So to develop systems to manage those risks and diseases in each province. And that's the next step. Working with those, with the tech transfer team, I think is going to be very valuable, because we can use that data, the results we get, and different provinces may have different issues or different risks that they need to manage. So that's what we want to target, and that's all. So if you guys have questions.